not very good. Can you turn the volume again?
Oh. It's the highest. Volume is the highest.
Maybe take off your mask, huh?
I'm kind of worried.
The virus will transmit it through the internet.
Upstairs in the fifth floor. Sixth floor already have one case.
Oh, okay.
Yeah. The sixth floor.
So we have how many? We see.
Now we still don't have that many people. We still have few examiners not in yet.
Yes.
Henry not come yet. And Robert Chi not come yet.
We have WK1, Bob Chi, David Wan, and Henry Lam.
So Kenneth, did I attend your qualifying exam, right?
What?
Did I attend Kenneth's qualifying exams?
Yes.
1 year ago or 2 years ago? When was your qualifying exam?
Kenneth.
Yes.
When was your qualified exam?
Last year, December.
Oh, last year, okay.
Hello. Hey, Professor Wan. Hello.
Hello.
Hey, David.
Hello, Professor Wan.
Hey, David, turn on your audio.
Hello.
David, turn on your audio to see whether we can hear you.
Hello, Professor Wan. Could you turn on your audio?
聽唔到你講嘢啊 ，David，嗰個音響係而家 OK，講啊。
Okay, is it okay?
聽啲。
Is it clear enough?
太細聲。
Well. Try.
Okay, we need one more question.
喂 ，WK。
你好。
好耐冇見。好久沒有見面咯。Henry's here, huh?
Yeah.
We are waiting
Hello.
for Bob Chi.
Robert Chi.
Hello, Harry. Hello.
So for the student, I do recommend you turn on your video.
Yeah, it's better. Turn on the camera.
Yes. So turn on your video, please.
David， 好耐冇見。
喂，你好，你好。WK， 別來無恙嗎
？OK 啦。
嚇？大家都
Okay, now we see Robert Chi.
Yes. So Chairman, we are all here now.
Okay, good.
Oh, Rochi, the professor is here.
Oh, we can start it?
Yeah.
Okay, good. Good afternoon, everyone, and welcome to the PhD Defense Seminar by King Leong Wan. I am Rong Biao-tong from the Department of Chemistry and will chair this PhD examination. Let's see, I have a pop-up. Okay, following the university policies, the candidates will present his PhD research work in 30 to 50 minutes. And then a Q&A section will be followed. And I would encourage audience from the floor to ask questions. And then the member of the committee will ask questions in a closed door fashions. And finally, we're going to announce the results after we have a full discussion and approval by all the members of the thesis committee. Okay, now Mr. Kwang, you can present your work.
Okay, thank you. Good afternoon, everyone. Thank you so much for coming to my PhD defense. I am Kenneth from Professor Carl Jim's lab. Today, I would like to share a story about the mass spectrometry-based multi-omics technology in resolving the action mechanism of herbal medicine. Traditional Chinese medicine has been utilized for thousands of years. Along with the history, the traditional Chinese medicine has been a special system to mitigate the illness. The source of the TCM is broad. It could be derived from flowers, the seeds and fruits, even the whole plants. The different herbs are mixed together that have been provided perform the better biological function. However, there is a lot of challenging of the TCM research due to the multifunctions, synergy, multi-pathway, multi-targets of a pharmaceutical function, as well as complex constituent of the herbs. In our herbal study, we chose the Dangui Bushetong DBT as an example of the study. This formula was created by the formulation pharmacist Li Dongyuan, the two herbs, astrogalic ready AR Wang Qi, as well as angelica synthesis ready Dangui. And the ratio is 5:1 is extracted in water. The clinical use is for women disease, such as the menopausal disorder, or other disease, such as cardiovascular disease, by enrich the qi and enrich the blood. In order to moderate the DBT, we construct a lot of biological research to solve the function of this formula, such as the preparation method and mechanistic analysis. However, some backbones of the DBT cannot be solved. For example, what's the low-reach the qi? What's the inherits the hot attribute of the DBT? As well as what is the relationship between the DBT and these biological active compounds in synergistic effect? Our previous research still cannot answer this question. Systematic biology is a holistic approach to resolving the complexity of biological systems. It gives a new insights, new hypotheses, and new biological questions to different areas, such as COVID-19 and developmental biology. Nowadays, multi-omics technology is important in systematic biology. The omics is powerful tools that can measure large family of biomolecules. For example, in our study, we will use the mass spectrometry to detect a huge amount of lipids, proteins, and metabolites. Today, I would like to derive the presentation in 3 parts. The first part is how we use the metabolic flux analyzer to solve the qi or energy metabolism in DBT. Second, how we define the multi-omics peptide to solve the relationship between the active compound and DBT. Finally, how we define the serum metabolomics and lipidomics approach to resolving the hot inherent attribute in DBT. The first part is effect of DBT in mitochondrial bioenergetics. In TCM theory, qi stimulate action is generally involved in enhancement of the body functions, and the effect is believed to perform by providing the energy. Based on this assumption, a lot of research conduct a lot of research about the qi, which is they conclude that this is associated with the ATP production, ATP-related enzyme, mitochondrial bioenergetics. Therefore, we can define that the action of the qi is equal to energy metabolism. The first finding of the DBT related to energy metabolism is in this paper. The author would like to find the defensive expression genes compared to DBT and control in genomic data. You can see that the more upregulated genes is highly associated with the energy metabolism. However, due to the technology is limited at this time, now we would like to use the novel technology to further study the energy metabolism of DBT. Here, we'll use the high effective tools to help us to get a comprehensive profile of the mitochondrial bioenergetics. By using the inhibitors of the electron transfer chain, the mitochondrial respiration could be controlled. The process is by injecting this inhibitor into the cell according to the time programming. The change of the oxygen consumption rate could be recorded. Then the key parameters of the mitochondrial bioenergetics, such as the basal respiration and ATP production, could be calculated accordingly. The H-like-C2 cell cardiovascular cell model was used in our study because it is well-defined cell model of the mitochondrial bioenergetic and energy metabolism study. By treatment with the DBT as well as other herbs, we gathered a comprehensive view of the effects of DBT to mitochondrial bioenergetics. All extras show the metabolic effect in dose-dependent manner. The DBT process the strongest metabolic effect to basal respirations, protein-like ATP production, and non-mitochondrial respirations. It implies that the DBT can increase the mitochondrial bioenergetics compared to other herbs. Moreover, the tone effect of the DBT to weak respiration cell model was also studied. By treated with the TPHP, the mitochondrial respiration could be inhibited. However, DBT could effectively restore the mitochondrial respiration by increasing the basal respirations, protein-like, and ATP production. So further explore the reason of the effect of DBT, the weak mitochondrial respirations. The 5 major compounds of DBT was selected to determine the special effect of this compound. From the result, you can see that the acetylglycide 4 possesses the strong metabolic effect to basal respirations, ATP production, maximum respirations, and spare capacity. Meanwhile, the lingostilide possesses the strong protein-like effect. We detected the mitochondrial membrane potential to explore the mechanism of the mitochondrial bioenergetics. The mitochondrial membrane potential is very important in electrochemical force to facilitate the ATP production. You can see that the DBT can get a larger stability in mitochondrial membrane in health cell model and weak respiration model compared to other herbal treatments. Moreover, we would like to see the effect of herbs on mitochondrial contact levels. Averagely, you can see that the DBT can increase the mitochondrial bioenergetics related genes such as the PCG1 alpha and mitochondrial DNA replication related genes such as the POLG compared other herbal extracts. It represented that the DBT can reduce the mitochondrial biogenesis as well as mitochondrial DNA replication. Finally, we would like to see the effect of the herbs on mitochondrial morphology. We use the mitochondrial web, which is the signaling dye specific for mitochondria, and observe under the confocal microscope. First, you can see that the DBT can most of increase the mitochondrial web intensity compared to other herbs. It implies that the DBT can induce the mitochondrial contact levels, which is match our previous results. Then we know that the high elongation and lower mitochondrial circularity are highly associated with oxidative phosphorylations. You can see that the DBT can trigger the parameter and reduce the circularities compared to other herbs. Meanwhile, you can see that the result was validated by electron microscope. It can represent that DBT can improve the mitochondrial morphology. In the part 1 summary, we conduct a comprehensive analysis of the effect of DBT on mitochondria, including moderate the mitochondrial bioenergetics, stability, the mitochondrial membrane potential, upregulate the mitochondria biogenesis genes and improve the mitochondrial morphology. Overall, the results show that Qi is highly related to mitochondrial bioenergetics as well as biogenesis in DBT. The second part is integrated omics dissect of the butterfly and boring effect in DBT on cultured osteoblasts. In our lab, we discover three effects in DBT. The first one is the synergistic effect. The second one is the boring effect. The third one is butterfly effect. I will explain it later. Synergistic effect in DBT. Synergistic effect of the herbal medicine is very common in TCM. Two or more herbs compared to each other will maximize the pharmaceutical result compared to single herbs. In previous result, you can see that the DBT has synergistic effect, the anthropolytic, osteogenic, and estrogenic effect. Boring effect means that the pharmaceutical effect of the herbs was increased after boring process. In a previous study, the pharmaceutical effect of DBT was compared to AR plus ASR. Here, the AR plus ASR means that the mixture of the AR and ASR extract in 5:1 ratio without boring. The ratio is same as the DBT. We can see that compared with just DBT, the AR plus ASR show a less increase in different functions, and AR plus ASR show the less increase in osteogenesis. In our lab, the first part is the butterfly effect in DBT. In our lab, we chose the glycosin as a lockout target compound for investigating the relationship between the DBT and active compound, because the glycosin is the highest abundant active compound in DBT. To generate the chemical DBT, we will utilize the semi-propagative high-pressure liquid chromatography, where the target fraction is collected. Next, we will use the glycosin lockout DBT to test the different biological function, which is reported from DBT. We can see that when compared with just DBT, locking out glycosin from DBT show a less increase in different function. We can find that compared with just DBT, locking out glycosin from DBT show a less increase in bone osteogenesis. Interestingly, the glycosin alone cannot induce osteogenesis compared with control. Overall, the osteogenesis function was chosen in our study later. The butterfly effect is like butterfly effect. The glycosin can regulate the DBT system. However, the DBT system will mess up our contributions without glycosin. But how to solve this mechanism of this effect? Because the one-way study in the synergistic effect is well studied. Here, today, we will employ the multi-omics as our major approach to solve the action mechanism of the butterfly and the boring effect. First, the osteoblastic functions was treated with DBT and glycosin lockout DBT. The total RNA was extracted and the RNA seq was utilized to find the differential expression RNA after treatment. When we are looking for the miRNA analysis, we found that such as the 337 miRNA, which is highly associated osteoblast differentiation, was significantly changed. Next, in mRNA expression pathway analysis, the circle size is represented the degree of significancy of the upregulated pathway. You can see several pathway classes was influenced by lockout DBT, including the RNA, amino acid, lipid, energy, and immunometabolisms. Also, you can find that some pathways which is not reported before, for example, the transcriptional regulation by small RNA and the response to the interleukin-3 in immune response. In addition, we also find the estrogen-dependent gene expression in our study, which is consistent with our previous result. Overall, compared with just DBT, the locking out glycosin from DBT show a less or loss increase in different pathways. Interestingly, we cannot find any pathway which is directly related to bone formation from defensive expression RNA in DBT or locking out glycosin from DBT. Next, we would like to examine the change of protein by locking out the glycosin DBT. We would like to perform the MS-based proteomics analysis. In the protein expression pathway analysis, the circle size is represented to the degree of significance of upregulated pathway. In the protein level, you can see several pathways was influenced by the lockout DBT, as I mentioned before in RNA level. For example, the tryptophan metabolism in glucose and amino acid metabolism, and the alpha ethylene acid in the lipid metabolism, and structural constituent of the ribosome in RNA metabolism. However, we can find lots of pathways are directly related to bone development. For example, the actin filament, the collagen biosynthesis and modifying enzyme, wing signaling pathway, and extracellular matrix receptor interaction. For other response related to bone formation, such as the zinc and magnesium ion transmembrane transporter activity also be found. Overall, to compare with just DBT, the locking out glycosin from DBT also saw a less or loss increase in different pathway in protein level. We would like to see the relationship of the defensive expression protein compared with just DBT and locking out glycosin from DBT. The level analysis was employed. We can see that the new information about what happened in biological system. For example, you can see the launch and AKT pathway, which is related to bone differentiation with activation, and alpha kappa B pathway, which is related to inflammation with downregulations, and the AMPK pathway, which is related to energy metabolism with activation. Post-translational modification is important component in cell signaling. Therefore, next, we would like to examine the change of the PTM proteomics by lockout glycosin DBT. Here, we're looking for the two most common found modification, phosphorylation and acylation. Then we just found that the lockout glycosin DBT affect the more in acylated protein is much than the phosphorylated protein. In the pathway impact analysis of the acylated protein, you can see that the lockout glycosin is less of the increase compared to DBT in some pathway. For example, the collagen trimer, the osteoblast differentiation, and actin cytoskeleton, which is related to bone formation. As I mentioned before, compared with just DBT, the locking out glycosin from DBT saw a less increased lipid metabolism. Therefore, we would like to further to study it, and we like to perform the MS-based untarget lipidomic analysis. We use the positive and negative ionization to aggregate the different chemistry of the lipids. In OPLSD model, you can see that the DBT and locking out glycosin from DBT has a clear separations. Next, we will use the S-plot to find the features which can differentiate these two experimental groups. The features was identified by the database and the manual inspection, such as the MADLIN and the lipid map. You can see that the defensive expression lipid can categorize in two groups. This implication of the lipid change I will discuss in part of proposed mechanism later. In the metabolic level analysis, we can find that a key lipid, the sphingoside 1-phosphate, is affected by lockout glycosin. And this is very important lipid to moderate the effect of estrogen in human osteoblasts. Moreover, we'll use the concatenation-based integration. The RNA, protein, and lipid data was integrated together, and we performed the pathway analysis. The first purpose of the integrated omics is consolidated with our result of single omics. You can see that the RNA, lipid, energy, and bone development has been significantly changed which is mentioned in single omics before. And second purpose is integrated omics is to find the low-flow network. It means that some network can be found in integrated omics, but cannot find in single omics. You can see some low-flow pathway interaction. For example, the activation of the osteoblastic divided VEGF, which can regulate the osteoblastic differentiation and bone formation during the bone repair. For the activation of the hypoxia-induced factor, which can restrict the osteoclast by the launch pathway in bone formation. Overall, we would like to propose the action mechanism of the glycosyl regulated DBT based on the omics data. I'd like to simply to say the first glycosyl can induce the glucose influx and oxidative phosphorylation to enhance the energy metabolism. Meanwhile, the fatty acid was utilized to beta oxidation. Some fat and also some lipid, for example, the phosphatidylate acid is utilized to the bone differentiation or proliferation. For other one, for example, the phosphatidylcholine and phosphatidylamphetamines was reduced the concentration to reduce the ROS stress. And then the ancosanoids can trigger the PGC1 alpha, and PGC1 alpha is very important protein for the mitochondrial DNA transcription and electron transfer chains. And finally, the hypoxia response element can induce the activation of the osteoblastic derived VEGF and BDGF for osteoblastic proliferation and differentiation. As we know that the AR and ASR was bound together to form the DBT. Compared with AR plus ASR, DBT can more increase the osteogenesis effects. Here, we would like to use the omics to rewrite the protein and lipid change compared to the DBT and the herbs extract without the bonding together. In the pathway analysis of the differential expression protein, generally, you can see that compared to DBT, AR plus ASR show the less or loss increasing in bone development, RNA, energy, and lipid metabolism, as well as other response related to bone formation, such as estrogen and zinc iron binding. However, we cannot find any pathway related to immune response from differential expression proteins. Overall, this pathway, which is less increased in AR plus ASR, is very similar to knocking out glycosyl from DBT, maybe due to amount of DBT was increased during the bonding process, which is reported in previous study. In the lipidomic study, we will use the positive and negative ionization method to acquisite different chemistry of lipid classes. You can see that the DBT and AR plus ASR has a clear separation in OPL state model. Next, we use the S-part to find the features which can differentiate these two groups. And you can see that the differential expression lipid can be customized into two groups. Interestingly, the AR plus ASR and glycosyl knockout DBT have same upregulated and downregulated lipid compared to DBT. In the next part, we will use the cellular physiology to validate our omics result. First, we employ the free assay to test the bone formation on primary cultured osteoblasts. The first one is RUNX2 promoter assay, is key transcriptional factor for the bone formation. The second one is proliferation assay, and the last one is ALP, the alkyl phosphatase, which is one of the bone differentiation marker. You can see the glycosyl alone in the DBT amount cannot induce any bone formation significantly. However, in terms of the bone formation and differentiation, glycosyl knockout DBT as well as AR plus ASR show a clear increasement when compared with DBT. Collagen synthesis and adhesive capability is very important in osteoblast development. Here, we can see that compared with just DBT, the locking out glycosin from DBT and AR plus ASR show a less increase in collagen synthesis and the adhesive capability. The osteogenic effect of the herbs was demonstrated in the ex vivo model. The mineralization process of the micromass culture indicated the direct by allicinated red staining, and the corticogenesis indicated by alkane blue. You can see that compared with just DBT, the locking out glycosin from DBT and AR plus ASR show a less increase in osteogenesis in ex vivo culture during to DBT can induce more allicinated red compared with two groups. Here, we would like to employ the vital mass assay and the glycolysis stress assay, but treated with DBT as well with other herbs. We got a comprehensive view of the effect of DBT to glycolysis and phosphorylations. The DBT can process the strong metabolic effect to basal respiration, ATP production, maximum respiration, spare capacity, glycolysis capacity, and glycolysis reserve. This is implied that the DBT can increase the mitochondrial bioenergetics and glycolysis compared to other herbs. We would like to see the effect of the herbs on mitochondrial dynamic, which is introduced in chapter in part 1. Then below that, the high elongation and the conductivity are highly associated with the oxidative phosphorylation and enhance the fusion process. You can see that the DBT can more trigger the footprints, the bench length, and network bench compared to other herbs. It can represent that the DBT can improve the mitochondrial dynamic. Here, we would like to employ the electron microscope to see the osteoblastic mitochondria after treatment. High elongation mitochondrial density crystal area are highly associated with oxidative phosphorylation and enhance the fusion process. You can see that in general, DBT can induce more increase in mitochondrial length, the crystal area, the mitochondrial area, and the density in the cell compared to other herbs. Let's get the sub-conclusion. We demonstrate the in-depth and integrated peptide. For example, the chemical lockout, cellular physiology, and integrated omics approach to reveal the activation glycosin of DBT on cultured osteoblasts. Reduce the osteoblastic function, proliferation, or differentiation of the glycosin knockout DBT and AR plus ASR in cultured osteoblasts compared to DBT. The osteoblastic function of the glycosin knockout DBT and AR plus ASR is not related to glycolysis, energy metabolism, lipid metabolism by integrated omics approach. The results suggest that the glycosin can engineering control the osteoblastic function at the pathway in DBT naturally and spontaneously. In the part 3, we will use the serum metabolomic and lipidomic to reverse the thermogenesis of astrogalic ready on yeast-induced animal model. The influence of the chi herbs to the energy metabolism would result to increase the body temperature. This is the traditional Chinese field. The part 1 and part 2 result, you can see that the DBT can facilitate the energy metabolism in cell model. Also, DBT can trigger some pathway, such as the beta oxidation, which is related to heat generation. Instead, could conduct the heat stimuli and watch the action mechanism. Here, we will choose major chi herbs in DBT as astrogalic ready for further study and apply it to in vivo model, animal model, in order to see the hot stimulating effect in the whole biological system. Here is the yeast-induced parousia rat model was introduced as the systematic performance of the animals induced by yeast is very similar to common information. This model is commonly used to investigate the anti-pyrotic effect of the drugs. Here, the several groups, including the control, the model, and positive control, and astrogalic ready, as well as the free herbs, was designed. Apart from the control group, the other group was induced by yeast, and the body temperature was monitored every 2 hours, and the serum was injected to MS for metabolomic and lipidomic analysis after the sample preparation. Here is the detail by administered with the herbs and aspirin 1 hour and 3.5 hours after injection of the yeast suspension. Then the temperature was measured every 2 hours. We can see that the temperature of the model mouse saw a significant change compared with the health mouse. And the aspirin, as a positive control, could suppress the rise of the temperature rapidly. Different from this medicine, cockles showed a significant cooling effect since 10 hours and opposed the temperature in astrogalic retic group are even higher than the model group. Overall, this result are consistent with the herbal nature attribute of these herbs. Meanwhile, we found that the 8 hour is the inflection point of the temperature change. Furthermore, the effect of these herbs to fatty acid were studied. With the gas chromatography mass spectrometry, the active free fatty acid and active acidified fatty acid in serum were identified and quantified quantitative analysis. Similarly, we found that this free fatty acid could also be derived in 2 groups. One is the positive correlated with the body temperature, such as the model, astrogalic and ginseng radius, and other one is negative related, such as the Leland bias and the cockles serum in PLSD model in 8 hours. It implies that hot and cold herbs can significantly change the fatty acid level in serum. To see the differences of the lipid exchange after the hot and cold herbs treatments, we will use the heat map to visualize the quantitative analysis of the fatty acid. You can see clearly the hot stimulating herbs, the general reduce the fatty acid and acidified fatty acid, but the cold stimulating herbs is opposite. In addition, we would like to further explore the effect of these herbs to lipid were studied in 8 hours. With LCMS, some lipid in the serum were identified and quantitative analysis. Here, the QC sample, poor sample, is used for the qualitative control. Similarly, we found that this lipid could be also divided into 2 groups. One is positive correlated with the body temperature, then other one is negative related in the PLSD model in 8 hours. To see the difference of the lipid change after the hot and cold herbal treatments, we will use the heat map to visualize the quantitative analysis of lipid. You can see clearly the hot stimulating herbs are generally reduce the triacetylglycan and sprinkle mildly, but the cold stimulating herbs is opposite. Next, we would like to give effects of these herbs to different classes of the metabolites with LCMS approach. Here, the QC sample is also used for qualitative control. Similarly, we found that this metabolite could also derive in 2 groups. One is hot stimulating herbs and other one is cold stimulating herbs in PLSD model in 8 hours. It implies that hot and cold herbs can significantly change the different classes of the lipids level in serum. To see the different metabolite change after hot and cold treatments, we will use the heat map to visualize the quantitative analysis of metabolites. 96 metabolites was identified by the LCMS metabolomics. We can see some metabolites, such as the keratins, and show positive relationship with the regulation of body temperature. When the level of amino acid and bile acid were negatively correlated to the temperature change at 8 hours, integrated with our findings from the lipidomics as well as the fatty acid analysis, we can imply that the hot and cold stimulating helps change the biological molecule, which is related to immune response, and thermogenesis, which is associated with energy metabolism. For my conclusions, the yeast-induced information read after treat of the cold and hot herbal medicine were used as our model system. The multi-omics approach identified the clustering of the cold and hot treated rat serum by using PLSD model. The levels of the biological molecule in serum, such as the phosphatidylcholine, show the positive relationship with the regulation of body temperature. When the level of the triacylglycan and sphingomycin were negative correlation to the temperature change at 8 hours, which is related to thermogenesis and immunoactivation. Overall, we provide strong science-based or insight to reveal the pathway changes by hot and cold stimulating herbs. For my conclusion, we will use and derive the low-flow, robust, and high-throughput MS-based integrated omics platform. For example, the MS-based proteomics and metabolomics. This platform can contribute to our understanding of the primary mechanism of DBT, such as the energy metabolism and the thermogenesis. Also, our low-flow approach could serve as a critical backdrop for future study that characteristic the impact of the broad array of different factors of the herbal medicine. The investigation of the action mechanism in applying in multi-target drugs or combination therapy should be necessary for the modernization of the herbal medicine. For the acknowledgement, I would like to thank my supervisor, Professor Carl Jim, Professor Harry Lam, advisory committee members, the Professor Robert Chi, Professor Wen Chong, and defense examiner, Professor W.K. Wong, Professor Tong Bell, and Professor David Wang from CUHK. And also, thanks all the labmates, friends, and families. The questions are welcome.
Hello?
Okay. Now questions from the audience. So you either can turn on your microphone or you just type it. I try my best to figure out your questions.
Hello?
Hello.
Yes.
So I would like to know, like at the end, you chose 4 different herbs for your hot and cold stimulating experiments, right?
Yes.
So why these 4 in specific? There are a lot of different ones. So why these four herbs?
Okay. Okay, these 4 herbs, we'll choose the astrogalic ready and ginseng ready as a hot or qi herbs, because in the traditional Chinese medicine is to tonic the qi and increase the yangs. And also in the literature, the lemon bias serum and cortical serum is to represent the yin. In some scientific basis, we will, periods before, study before, we'll use the PCA. We have a classification, the yangs and yins herbs with hot or cold herbs. We find that in the PCA classification, these 4 drugs can be classification as separate in 2 clusters. So I will choose these 4 herbs as our further study.
Hello.
Hello.
I have one question. In your bone differentiation, you use the AALP assay to detect the alkaline phosphatase activity. So I want to know how to perform this assay and how to make sure that the specification of this specificity of this assay.
Okay. perform the ALP assays, because we will use it's a compound, is the substrate is called the PNPP. That when add the PNPP into the ALP lysate, they will break down the phosphates and will generate this compound. And this compound is is the yellow. So the solution color will from colorless change to yellow. So we'll detect the yellow color. The wavelength is the 405, can determine the ALP activity. And in the ALP assay, because in our cell assay, they have a acidic and alkali or neutral phosphatase. So in our LP buffer, we will specially decide the buffer pH is 10.4. So in this environment, the acidic and neutral phosphatase will be deactivated. And the second point, we will add the zinc concentration of the zinc chloride and the magnesium chloride to provide the cofactor to the alkyl phosphatase. So this assay is very specific for ALP.
Hello.
As you mentioned before, downgrade busulfan is for treatment of menopausal disease. Can men or does men or can men also use downgrade busulfan as a treatment? And my second question is why you choose male mouse in your serum omic experiment? Thank you.
So in the normal conditions, the man can use the DBT is a function of the herbs to lowish the qi and enrich blood and regulate the blood circulations. In our experiment, there's a lot of related pathway about the DBT is about the energy metabolism. So even the man or woman can use the DBT to increase your energy metabolism. Okay, for the second questions, okay, why we chose the man mouse? Because to prevent the metabolic changes in estrous cycles.
Hello. Hello.
Oh.
I would like to ask a question. I know that mass spec-based proteomics has two approaches, top-down and bottom-up. So which you use and why?
Okay, we will in our experiment we'll use the shotgun. It means that the bottom proteomics, and I'd like to compare the Okay, we'd like to compare the bottom-up and top-down proteomics. We have three important parts. The first part is the protein identification, and the second part is protein quantification. Because our study is on-target proteomics, it means that we'd like to compare the control or judgment to find the defensive expression protein. So if I can increase the protein identification and increase the protein quantification, so this approach is match our aims and study. So in here, the bottom proteomics, the number of the protein identification and number of protein quantification is much higher than the top-down proteomics. But in here, I'm not do any very precise about the post-translation modification. So overall, I will choose the bottom-up proteomics approach. Thank you.
Thank you, Kenneth. I have a question. Could you please explain how to analyze the metabolomic data and keep the quality control.
Okay, simply to say, for the metabolomic or lipidomic analysis, first we treat the cell was treated with your drug, and the drug will extracted by some organic solvents, and okay, the metabolite will injected into LC-MS, and the compound will identify the mass to charge ratio, and would like to know the structure of this compound. Will perform the MS, which means that you use the collision energy to break down the compound to see the fermentations. So the compound will identify, and the data the pit will process an alignment and will pass to the multivariate analysis. Use some model to identify the biomarker, compare with control and drug treatment. After identify the biomarker, we can compounds that we can identify this compound or this biomarker can use the database or manual expectations, and finally this compound will identify it and put it to pathway analysis. For the QC, I would like for my practice is use the QC pool sample in the before of the bench and after of the bench. What's the QC sample? The QC sample is the poor sample of each sample to add off the same volume of the sample to this is the poor QC sample. And in the bench, we'll put the QC sample in the before and after the bench injections. And the middle will also put the QC sample in the bench of the samples. And also in some injection position, we will put the reference standard to test the machine, for example, the intensity or retention time. Thank you.
Hey, Kenneth.
Yes.
Can you just go back to the previous slide? No, the previous one. Metabolite.
So for committee member, we can give a later on for questions.
Yeah, this is the one. So you're talking about metabolites here, right?
Yes.
So what do you mean? Can you define metabolites to me?
The endogenous metabolites. That is endogenous compound from the cell.
So are you talking about primary or secondary metabolites?
I've not mentioned about the primary and secondary metabolites. It just mentioned the endogenous metabolites in the cell.
So you include both of them, primary and secondary metabolites here?
You mean the primary or secondary metabolite by our drug, right?
Yeah.
No included, because I will use statistic to cut. Will take off these compounds.
To take off what?
The secondary and the primary or secondary metabolite of the DBT active compound.
No, I think that you are talking about primary metabolites here, right? Not secondary. You know primary metabolites? Do you know the definition for primary metabolites?
Primary metabolites means that I would like to clarify the question. You mean the primary metabolite is coming from the drug or coming from the cell?
I'm just asking you, do you know the definition for primary metabolite?
Some metabolite is not modified.
It's not what?
Modified, or any biotransformations.
Does it have anything to do with growth of the cell?
Yes. Something is yes. Some metabolites, yes.
And energy consumption, right?
Yes.
So in here, I think that you're talking about primary metabolites, not secondary.
Yes, I'm talking about primary metabolites.
Okay.
Any other questions from students? So we can before we go to the closed sections for committee members. If not, then please excuse us. Leave the room. I think the host can make them leave, and so we can jump to the next sections. The committee member can ask questions.